So for that you don't need too much protein compared to the other tissues. So the ratio is 0.23:1. Now if we look at the composition of cell membrane, you have the lipid bilayer. So you have the outer leaflets and the inner leaflets, and mostly the lipid is very important over here. I told you that outnumbered by the molecules of protein by numbers only, but by mass lipids are around 40%, 40-50%, and the most prominent lipid of all is phospholipids. Now phospholipids, if you look at them, they are these they are these things over here, which you've also studied about in school. They've got the hydrophilic and hydrophobic regions over there. The hydrophilic region is what is made up of by the head region of this molecule. It contains an alcohol and a phosphate over there. This makes the head region, and there are two tails over there, and this makes up the hydrophobic region. So you have your phospholipid head and tail. Now the tail is fatty acid tail. They are made of some carbons, around 14-17 carbons long, and joined end to end, and sometimes these carbons are saturated. So when they are saturated, the carbon, it's one carbon links to another, links to another, links to another. We know that. But sometimes what happens is you get a carbon with a double bond, an unsaturated bond. Okay, so this one makes a kink, what we call a kink in the fatty acid chain. So this kink allows, it happens because of the unsaturated bond. Okay, unsaturated. So what happens is it will allow for a little more space to exist between phospholipids. Instead of having them packed end to end next to each other, side by side, we are going to have a little space over here because of this unsaturated bond, and this allows for more fluidity, increasing more fluidity of the membrane. So your unsaturated fats, your polyunsaturated fatty acids, and your monounsaturated fatty acids, you get them from fish. Your arachidonic acid and all of these types of fatty acids, they have these kinks in their membrane. Linoleic acid, arachidonic acid, they all make up these kinked fatty acid chains, and cholesterol usually hangs out somewhere in between this kink also, acting as a buffer for fluidity. Remember, cholesterol is a buffer for membrane fluidity. We'll go into that later. So what happens is you have the more unsaturated bonds you have in the fatty in terms of fatty acid tails, you will have more kinks, and you will increase the fluidity of the membrane, and you will lower the transition temperature of the cell membrane, meaning to say that the temperature from where it transitions from gel state to sol state, remember this, it becomes lower. So you are able to have a more fluid membrane at a lower temperature, which is okay, which is good, which is effective. So that's what it is. And cholesterol doesn't allow too much of fluidity to form at low temperatures, and it doesn't allow too much of rigidity or sol or gel state to happen at low temperatures over there. So if you look at the phospholipids, the main phospholipids, again, they are different in terms of is it on the outer leaflet or is it on the inner leaflet. The outer leaflet has got phosphatidylcholine and sphingomyelin, and the inner leaflet has phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. This is how these phospholipids are distributed. Now, something to know is that phosphatidylserine can switch sides and actually go to the other side. Phosphatidylserine can actually flip to the outer leaflet, and this is done by an enzyme called scramblase. It scrambles the organization, you could think of it that way. So this is going to give a signal to the immune system to eat me. It's called an eat me signal. Okay, so here this is one example where it's not two-dimensional motion, it's three-dimensional motion, where one membrane is flipping to the other side of to the other leaflet over here. Okay, and this serves as a coagulant surface and allows also for blood clotting. So your phosphatidylserine, when it comes to the outer leaflet, it allows for blood clotting. Okay, it's a very important signal for blood clotting. And if you don't have enough scramblase in your body, you have a condition called Scott syndrome. Okay, where you get to bleed a lot. You get this hemorrhagic disorder due to lack of phosphatidylserine exposure to take place. So what is phosphatidylserine a signal for? For clotting to take place, and its absence causes absence of the enzyme scramblase causes Scott syndrome, which causes bleeding. Also, phosphatidylserine is a signal for apoptosis. Okay, so phosphatidylserine is the eat me signal in apoptosis. Then you have your phosphatidylcholine, which is on the outer leaflet. Let's talk about this. This is the most abundant phospholipid. Also, it's also important in making pulmonary surfactant. It's also known as lecithin. So that's one more thing. Okay, and phosphatidylcholine helps you in assessing the amount of fetal lung maturity. Okay, so what happens is when a woman is pregnant and you want to know if her baby is mature enough, like if she's gone into premature labor, you can take a sample of her amniotic fluid and see the lecithin to sphingomyelin ratio. And if the lecithin to sphingomyelin ratio is more than 2, that means the lungs are mature and this kid has a better chance of surviving if the labor or delivery takes place now, if he comes now. And then you have phosphatidylinositol. This is on the inner membrane. This is very important for signal transduction because there are these G protein coupled receptors that play a role in signal transduction, and phosphatidylsinositol gets converted to IP3 and diacylglycerol. It's one of the raw materials for making the second messengers that pass a signal on to the cell. So just remember this for now. As we go to those topics in more detail, we'll discuss this. And it also plays a role in anchoring certain membrane proteins called glycosyl phosphatidylinositol proteins. Okay, GPI anchored proteins. So glycosyl phosphatidyl inositol proteins. Then other than that, we have this one. Let's talk about sphingomyelin. It's in the outer leaflet, and it is again very abundant in the outer leaflet, and it's especially located in an area where you have something called lipid rafts. What are lipid rafts? So they are high concentrations of sphingomyelin and cholesterol, which play a role in transport across the cell membrane. You can check the transport across cell membrane chapter for that. And other than that, it's very important in the myelin sheath, and it makes the nerve conduction much faster. If you don't have the enzyme sphingomyelinase, then you end up getting a disease called Niemann-Pick disease. So what's it called? Niemann-Pick disease. Let me just write that down over here. Niemann-Pick disease, where you get the big liver and spleen and mental retardation, and usually those people die in early life. And you also have other things with like red cells. If you look at them with increased sphingomyelin content in their outer surface, they become something called acanthocytes. So this is where you have an abnormal red cell. Instead of the concave lipid layer which you see of the cell membrane, you have these acanthocytes. They have a funny shape, and this shape is because of this one, increased sphingomyelin content in the outer cell membrane area. It is seen in a condition called A beta lipoproteinemia. A beta lipoproteinemia. Right, and then after that you have your phosphatidylglycerol also. This is another phospholipid which is present inside phosphatidylglycerol. Let's just clear that up. Glycerol. So this phosphatidylglycerol, also known as cardiolipin, is a very important phospholipid, mostly in your mitochondria. Right? It's the signature phospholipid of the mitochondria, and it's abundant especially in the inner mitochondrial membrane, and it is involved in your production of ATP because it's involved with the mitochondria, and also with apoptosis signaling, because the mitochondria plays a role in signaling for cell death to take place. So this again happens because of the phosphatidylglycerol, also known as cardiolipin. There's something called Barth syndrome, where you see the enzyme which is involved in metabolizing cardiolipin, and that's called tafazin. Right, so what happens is, if this is the gene encoding tafazin is mutated, then you get a condition called Barth syndrome, where the heart gets affected badly. You get this dilated cardiomyopathy where the heart becomes really big, it's dilated over there, and you get skeletal muscle weakness and growth retardation. So you get weak muscles, so skeletal muscle weakness. You get reduced growth, and you also have low neutrophils, neutropenia. So dilated cardiomyopathy, weak muscles, low growth, neutropenia. This is called Barth syndrome, B-A-R-T-H syndrome. Okay, be aware of this. It's X-linked recessive condition and only affects the males. Another thing about your cardiolipin, also known as phosphatidylglycerol, is if you look at it, there's a condition called lupus, autoimmune condition, where you get anti-phospholipid antibodies against this, or anti-cardiolipin antibodies. And people who have this condition, they are not able to carry a pregnancy to the end. Usually these they go into abortion. They lose the baby very fast. They get recurrent arterial or venous thrombosis also, and abortions of the child, because this happens in the pregnancy time, and this will cause death of the child, of the fetus. It is diagnosed also in syphilis, these anticardiolipid antibodies in syphilis, for venereal diseases, meaning by using the VDRL test, we are able to diagnose if a person has syphilis, and our anticardiolipin antibodies are seen in that condition over there. Right, so these are your phospholipids. We've gone into a fair amount of information regarding phospholipids.